Section 6. Key lessons from estimates of positive predictive value in germline next-generation sequencing tests. Section 6.1. Modeling positive predictive value to include both analytical and variant classification test characteristics. Sensitivity and specificity are familiar terms when describing diagnostic test characteristics. Mathematically combining these test characteristics with the true genetic disorder prevalence in the population being tested provides the clinical positive predictive value, or PPV, of the diagnostic test, as defined with the standard equation shown in Figure 2a. In the modeling demonstrated here, clinical PPV is the percentage chance that a patient's variant, when reported by the laboratory as positive, is truly positive. Positive variants are those classified as pathogenic or likely pathogenic. At present, most genetic and genomic sequencing test reports that have incorporated variant classification are accompanied by technical specifications that describe the test coverage and technical limitations of the test, including metrics of analytical sensitivity and specificity. These analytical metrics relate to the performance of the sequencing instrument and the variant calling pipeline. Test specifications rarely, if ever, include metrics of novel variant classification sensitivity or specificity, since variant classification lacks the public truth sets needed to generate classification sensitivity and specificity metrics. Therefore, in the absence of variant classification sensitivity and specificity estimates, the clinical PPVs of most modern genetic and genomic NGS tests have been left unexplored until now. Two of the authors, Giles and Eggington, created a framework to model clinical PPV in NGS germline sequencing tests that incorporates both the reported analytical performance of the tests and informed estimates of the variant classification sensitivity and specificity. The framework of inputs and outputs is diagrammed in Figure 2b. For simplification, the 5-tier germline variant classification system was converted into a binary system based on the most common clinical scenario, where variants classified as pathogenic and likely pathogenic are likely to alter clinical care, positives, and variants classified as likely benign, or VUS, are not likely to alter clinical care, negatives. In the simplified model, a germline genetic disorder is caused exclusively by autosomal dominant simulated genes and is entirely detectable within a simulated 100-gene diagnostic panel. The hypothetical gene sequencing lengths are evenly distributed between 14.5 kilobases and 1.1 kilobases, modeled after the diagnostic sizes of the large ATM gene and the small CDKN2A gene, respectively. A hypothetical gene is randomly selected to capture 30% of the genetic disorder and represents the single gene test scenario. The expanded 20-gene panel with additional randomly selected genes captures 63% of the genetic disorder, and the 100-gene panel captures 100% of the disorder. The NOMAD data for the large ATM gene is used to set an average rate of variation of 0.32 variants per kilobase in the model, with each allele contributing 0.16 variants per kilobase. While variation is not evenly distributed across the genome, this assumed constant rate of variation serves as an acceptable basis for a simplified illustrative model. The simulated autosomal dominant genetic disorder is set as having a general population prevalence of 1%. The analytical sensitivity and specificity of the simulated 20-gene panel for high, medium, and low stringency laboratories is set to 99.99% sensitivity and 99.99% specificity for high, 99.95% sensitivity and 99.95% specificity for medium, and 99.00% and 99.00% sensitivity and 99.00% specificity for low, to match laboratory-reported test characteristic ranges for real clinical laboratory sequencing tests of somewhat similar size. These analytical test characteristics are then extrapolated to the single gene and the 100 gene modeled tests. The rate of novel variant classification true positives, false positives, true negatives, and false negatives is estimated from published counts at the patient level of initial variant classifications and then reclassifications in hereditary cancer from a major laboratory over a 10-year period. For example, pathogenic and likely pathogenic classifications that were later downgraded to VUS, a positive to negative reclassification in our model, are categorized as false positives, while pathogenic and likely pathogenic classifications that were not reclassified during the 10-year study are considered true positives. Although reclassification counts in this 10-year study cohort will increase over time, the data come from a laboratory that has consistently operated a proactive variant reclassification program. The laboratory's published classification and reclassification counts therefore serve here to optimistically estimate the classification, sensitivity, and specificity of a very stringent genetic test, according to the standard equations illustrated in Figure 2a. Such an analysis of these data result in a novel variant classification sensitivity and specificity of 98.5% sensitivity and 99.995% specificity. Based on these estimates, we model the variant classification, sensitivity, and specificity for high, medium, and low stringency laboratories at 98.00% sensitivity and 99.99% specificity for high, 99.00% sensitivity and 99.98% specificity for medium, and 100%, 100.00% and sensitivity and 99.97% specificity for low. Note that higher stringency laboratories gain specificity in part by decreasing sensitivity, which in practice typically means requiring higher levels of evidence for variants to be classified as pathogenic or likely pathogenic instead of as a VUS, as compared with lower stringency laboratories. Variant classification, sensitivity, and specificity, as treated here, are at a per observation level and not a per unique variant level. Each variant observation is treated independently. For example, if there are two unique variants, variant A and variant B, variant A is a false positive, while variant B is a true positive, and in 10 patients, variant A is observed 1 time, and variant B is observed 9 times, then 1 out of 10 variant classifications is a false positive, and 9 are true positives. This patient-centric approach is why the classification specificity estimates used here appear very high to variant classification scientists accustomed to wrestling with the evidence of only rare variants. The cumulative allelic frequencies of the variants that are very common, and therefore very easy to accurately classify as benign, contribute a high observation count of true negative variants to the specificity equation. The cumulative allelic frequency of a few common variants often exceeds the cumulative allelic frequency of many rare variants by more than an order of magnitude, as exemplified when using an arbitrary but realistic threshold in the ATM gene in Figure 3.